Hello to everyone. My name is Darius Kavaliauskas, and I am going to present the guidelines for conducting genetic monitoring in the field for Abies alba and Abies borisii-regis. This guidelines was prepared in the frame of the project LIFEGENMON, and it shortly describes silver fir and king boris fir, their reproduction system, environmental requirements, and threats. In addition, they provide guidance how to establish, select, and to maintain forest genetic monitoring plots and how to record all the field verifiers. These guidelines are based on actual results from LIFEGENMON test sites. And as you all probably know well, silver fir and king boris fir are wind-pollinated, monoecious, generally outcrossing conifer tree species. Seeds of both species are wind-dispersed, and silver fir is one of the most important forest tree species from economic and ecological point of view in several European countries. And king boris fir is an important natural hybrid between Abies alba and Abies cephalonica, growing mainly in Greece. Both species are facing many threats and challenges due to climate change, and therefore they are both considered for forest genetic monitoring. So silver fir is a stand-forming tree species, therefore regular forest genetic monitoring scheme should be applied. In contrast, the genetic monitoring of Abies borisii-regis due to geographically distinct distribution, biology, for example, here hybridization and other threats, can be a bit more challenging and time-consuming. However, when selecting forest genetic monitoring plots for Abies, it is important to contact and to have some consultations with local foresters, since Abies can appear in the form of groups among the other trees in mixed forest stand. It is good to know where it is present and what is the density of the trees. In addition, orthophotography can help, since the Abies is clearly visible and distinct, and it is easy to distinguish from other tree species. It can help to see in the area where it is present. And also at the end, the initial field survey should be performed. It means that we should go to the plot to check the actual number of trees, to see the distance between trees, if the number of trees is sufficient, and if there is enough of natural regeneration. So since the plot for genetic monitoring is selected, it follows the plot establishment, and the design of genetic monitoring plot should be contain 50 mature reproducing trees distributed over the forest stand, and the distance within among the trees should be at least 30 meters. In addition, DBH or social classes can be used as a proxy to identify reproducing trees if the plot is being established outside of the flowering season. And the last but really important condition is the presence of sufficient density of natural regeneration in genetic monitoring plot. So Abies alba guidelines, as you can see, contains a list of verifiers and background information which should be recorded during field work. The genetic monitoring plot, and as you can see, all verifiers and background information are divided into 3 levels of genetic monitoring, into the basic level, standard level, and advanced. And the differences between those 3 levels, the differences are that the frequency and resolution of the data which we are collecting. These different levels are different. Yeah. So at the end, to be able to perform, for example, flowering or bud break, to collect the flowering or bud break data within the project LIFEGENMON, we have prepared a number of illustrations, which illustrates the different development stages, and it will be present in our guidelines. So overall, the guidelines provides the clear guidance how to perform genetic monitoring for Abies species.